strap a bunch of GoPros, and they're going to follow us out of the plane as we jump into amazing places. It's like Antarctica and Egypt and Chile and Peru and some other undisclosed locations, and take you with us on an adventure to show you, even into the Vatican as well, and take you with us to show you what really goes on behind the scenes, not so much sitting and doing just, talking to amazing people. We're going to show you how we get called upon to remote view certain sites and to also do excavations and gather some DNA from amazing creatures and bring them back to different universities. Yep. And bring them back to different universities for testing. And this is what we've been doing for quite a long time. And you don't hear about it in the mainstream. So we're going to take you on a beautiful adventure. And so stay tuned for that. And that's Zero Cross Productions. And that's going to be doing that. So, Olivia, what would you like to know about hybridization? And don't tell anyone about how I grew you or anything in the lab.
Why don't you start with your introduction to the genetic programs as far as cloning, hybridization, PLFs, and I'm sure you're all familiar with the autopsy stories. And what was your first introduction into hybridization and cloning? Was that after you did ET autopsies or during, or how long into the projects did it take you to get to that point?
It was probably about the first, that's a great question. It was probably like the first year into it that I started seeing people in the projects that looked exactly alike. And I knew, oh, well, maybe that's just a cool twin. And then I was like, there's no way. There's like 10 of those twins.
You mean coworkers or just
Yeah, these are coworkers. These are
so
coworkers.
You saw coworkers that looked exactly alike.
I never even spoke to you about this. Yeah, so there was actually coworkers in there that looked alike. And I know where you're going to go with that, by the way. But it was really interesting. But aside from that, there was many beings that I got on the table that looked like us, even, that were very similar, very identical, even, as far as the way they looked, just like you would see in a identical twin. And that's how I knew, this definitely, they're doing something here, growing these people, or 3D printing them, which later on I found out they were. The problem was the brain, it was the hardest thing to keep up. They could grow the tissue really fast. Tendons, ligaments, muscles, even the organs, but the brain had a problem keeping up with the rapid rate of acceleration. The reason we use, just so you guys know, we use pig heart valves in people is because the metabolic assimilation of the cells are at the same rate as humans. Isn't that amazing? So you can't just grab something and put it with something and it's just going to grow, because you have to be at that same genetic frequency metabolic rate. Now, however, if you get the DNA from a pig, porcine, and you mix it with human DNA, which they've already done in China, and people heard about the pig boy. I mean, they've been doing it for a very long time. That it actually bonds and does work. It works. And so now adding other different types of DNA to that, such as reptilian or equine or mammalian or what have you, they now have conquered that, and it's just a small percentage of the DNA from these other creatures on the planet that they conquered this now to make that genetic code. So they have mixed our human DNA with basically every living organism, including viruses and bacteria on the planet. Yeah. So
what are the side effects of hybridizing humans with all these organisms?
So we have a lot of problems with this. And the problems they were having was that anytime you grow anything in a petri dish that's not actually in a womb or inside some sort of body, you have a mutagenic problem where the cells don't always replicate and are exactly duplicates like they would be. And there was one of these creatures here, specifically, this is an artist's rendition, but it looks very similar to this, where they were crossing feline with human and pig. And this creature was coming out with, you know, a lot of dysmorphia. The skeletal system was not correct. And that you could tell the teeth were not correct. It was cross-eyed, and it was because they were interbreeding hybrids with hybrids. So they would make a hybrid. If it worked out, great. If it didn't, they would still save those cells and save that hybrid somewhere else to be tested on later on. But they would still use those cells and maybe make another thing. So they kept streaming all these different animals, these hybrid animals and people. Now, what had happened with that is they were testing that because they started getting a lot of extraterrestrial DNA and extraterrestrials that were coming here. Either they acquired them from spacecraft wrecks in the exoatmosphere, or they've shot them down and they've collected them here. And people are saying, well, you know, Emory was part of this project where they were harvesting all these. Yeah, it's not like that. They weren't just harvesting ETs. They weren't just, grabbing them and trying to grow them, by the millions. It wasn't like that. It was once in a while they would acquire a body, and then they would, of course, take that body and try to preserve it for as long as they could and do many, many tests with it. Many pharmaceutical corporations has used the technology from the DNA from the stars to help us, heal certain genetic factors. And now you're hearing about in science that they are, working on, we can do, gene sequencing where we can, find just one gene in there and we can manipulate that gene so you don't have to have this problem anymore. And that is true, and that's all because of the technology through what they have been doing for the last 30, 40 years.
So how do you tell the difference between, is there a huge difference, like physically, with human hybrids? I mean, do they all look like that tiger? Do some of them look just like you and I, or is that really more of a clone? How would you tell the difference scientifically and genetically and just by looking at them?
Well, I wasn't privy to a lot of the paperwork. I know you guys can't see me now because I got out of those lights, right? I don't like those lights. Bad light.
Good.
I'm very sensitive like you guys. I don't like lights and things around me. The EMF fields are just driving me nuts. I can deal with it. I just don't want to. So getting back to your question, how do they look? Is that the question?
Yeah, and how do you tell the difference? Like if you're analyzing the tissue, if you're dissecting it, is it more friable? Is there a difference with when you're working with it?
Well, the tissue is always different if it's grown in a lab. And anyone that's a lab person or a physician knows what, of course, healthy tissue looks like. And after you do this under a microscope forever, you pretty much know how the cells look and how they react and, the actual, how can I say this? The actual strength of the cell membrane is what we look at. And that lets us know how old the cell is and how well it performs and how long it's been dead. And some of these cells are actually still, on a lot of these autopsies we've done, even though the being is dead, it's still completely, looks like it just passed away within hours. So the cells have a great integrity, and the nucleus captures a frequency that we've measured that keeps the body in this stasis kind of thing. And they just learned this recently after I got out of the projects that, wow, we're actually operating on things that probably could be resuscitated, is what I'm getting at, if they were on their planet. So that was not the best thing. And so getting back to what they look like, well, they could look like many different things, but you can tell by the tissues as we're dissecting these creatures that they are definitely either grown, or they're cloned, or there's just something about the tissue that's, like she was saying, it's very friable. The pores of the skin are a lot larger because they're not used like our pores of the skin are. They might have facial features that, you know, are different than ours because they don't have to breathe. Some don't have to eat. They only have one esophagus that goes all the way down and out. And so you skip the stomach, the colon, and all those things, because these things only last for a couple of weeks, actually, a programmed life form or a clone. They have all that cellular material in there just like we have. They're just a little different, or there might be multiple things in that cell that are a little bit different. A couple more different organelles, in there that are different than ours, but they all have a mitochondria, a nucleolus, and a nucleus, and they all have, of course, the DNA, which is what they're after, which I realized later on, is basically they're testing the frequencies of these DNA and trying to morph it with human DNA to create a super soldier type being. And they were doing that with animals for the longest time. And there was one time on the base, well, that's really spooky. Oh my gosh. Come over here.
What's up?
I'm scared of that.
Yeah, why would they, what's the purpose of creating these creatures? What are they using for? Why would they even do this?
Well, they were trying to improve our longevity, number one, and they were also trying to figure out how they live so long.
What do you mean our longevity?
They live a lot longer because they have a lot better protection from the sun, from where they're from. And it's the radiation here that ages us.
Got to be lit.
There's a light here.
So they were studying. So you're saying.
Good morning. So yeah, what I'm saying, yes.
They were studying these beings to see what they could